Welcome, everybody. I'm Pat Levitt, I think. I was on the East Coast for the last 3 days, so I'm not sure what time zone I'm in anymore. I'm the Chief Scientific Officer at Children's Hospital Los Angeles and the Director of the Saban Research Institute, and it's a pleasure and an honor to welcome all of you to the Los Angeles High School Internship Program, and it's the annual Science Symposium. It's a summer symposium as well, right? You call it that? I was just talking to the brainchild behind HIP, with who you'll hear from later, Dr. Emil Bogenman, an oncologist by training, basic scientist. This program started around 20 years ago, so it's about the 20th anniversary. So we should give HIP an applause for the 20th anniversary. That was not in my notes, but I just decided I would say that. So my first research grant, well, my first grant from the National Institutes of Health, which was before almost everybody in this room was born, was a high school training program. I was in Philadelphia then at the Medical College of Pennsylvania. It was a high school program to have high school students from Central High School and Girls High School. In Philadelphia, they had Philly?
Girls High Girl.
Girls High Girl, right there. Oh my gosh. So you know I'm not making the story up. And you know where MCP was on Henry Avenue, right? So what I learned from that experience, one is bringing high school students to MCP to work in various laboratories from microbiology, immunology, neuroscience, pharmacology, et cetera. And part of my task was to talk to families about why this was so important, and I thought it was a great opportunity for their students. I work with the science teachers at Central and Girls High School about this. And what I learned from that is that while it was a wonderful program for the students, at least I think it was a wonderful program for the students, it was so important to have family support and family encouragement. It made a huge difference. So for all the families here, give yourself a round of applause because it really matters a lot. So you'll hear from the current director, Dr. Mark Fry, about the program. It's a research program. It's a professional development program. It's a program to recognize that great science is done in a social setting. Nobody does science on their own. There's a tremendous amount of interaction, and you'll see how your students have grown in just a relatively short period of time in terms of gaining an amazing amount of information and understanding of complex biomedical problems that are designed to solve the mysteries of pediatric disease. That's why we're here at Children's Hospital Los Angeles. As you know, this is one of the largest freestanding children's hospitals in the country. 750,000 children and adolescents come through there annually. It's a safety net hospital. We see all families, and we have amazing clinical and basic science programs that many of your students and all of you have experienced. So I do want to thank several individuals. They're on the notes that somebody gave me. And I want to thank, of course, Danielle Nunez and Emmanuel. And Emmanuel, where's Emmanuel Passon? Where is he? Where's Emmanuel? Emmanuel is okay. He's somewhere. Danielle's a program administrator. Emmanuel is a program coordinator. Been fantastic. You'll learn about the incredible faculty mentors. Mentorship is so important for me. It was very important. I was a work study student as an undergraduate, was able to go from glassware washing in the lab, that's my job, to doing experiments, and it was instrumental for me to make my career decisions about going into science. It was fantastic for all sorts of reasons that I know that many of you have experienced, having the good fortune of doing it, as you well deserve, in high school. The other acknowledgments, of course, is those who support this program. Started with the Samuels Family Foundation and Ted Samuels, who developed this amazing relationship with Dr. Bogenman about the initial gift that really jump-started this program, along with grants that Emil was able to get from various organizations. The O'Connell Family Foundation, Mary O'Connell, is a member of the Foundation Board of Trustees, who's been a longtime supporter of LA HIP. The Chuck Lurie Family Foundation, who, in addition to this and other programs, has provided a transformative gift to Children's Hospital of Los Angeles for all pediatric education programs here. Really quite amazing. And then the California Institute for Regenerative Medicine, we have a training grant with them, which is near and dear to my heart since I'm on the governing board of that. I had nothing to do with reviewing the grant, of course. So I think it's going to be an amazing day. I do have a meeting back across the street, which is the penalty I pay for whatever I'm supposed to be doing as the chief scientific officer. Mark, it's your time. Dr. Fry, Mark Fry, who, by the way, we overlapped and knew each other at Vanderbilt University, and Mark has done just an amazing job with all of you, and you're here to say more words. Thank you very much.
Thank you, Pat.
All right, thanks, Pat. I do want to just say thank you to Pat and to Jody and to Karen and all the folks from TSRI who've been really so supportive of the program for all this time. We wouldn't exist in the form we're in, of the size we're at, without the really strong support from the institution. So thank you for that. And I love going after Pat in the lineup because he's really good about thanking most of the people that we need to thank. So it makes my job that much easier. As he said, many thanks to the people who got the program running, including Emil, Ted Samuels, as well as the O'Connell Family Foundation and Mary O'Connell, and we have had great support from Chuck Lurie. I also think we need to thank the entire TechPad office, headed by Virginia Mason. That's where Emmanuel and Danielle are based. We also have Metzi and Brittany, who've been helping at the door today. Just the whole team is always there to pitch in to make things happen. I want to thank Roberto Archer over there in the AV booth, because he's the one who makes all this stuff work. And I've actually done that job in a much younger incarnation of myself, so I know how much of a pain it is, especially when you got people like me up here talking and moving around away from the microphone. And all of the mentors and the students and postdocs and technicians in the lab who have, it takes a certain kind of person to be willing to put in the time that's required to train someone from scratch to be as good as these students are in the lab now over the course of 7 weeks. And every year our mentors just do an amazing job, and I'm always astonished at what the students in the program are able to accomplish in 7 weeks, from most of them walking in never having seen a pipette, never having been in a room with a real working professional microscope, et cetera, and then they're really truly well-trained scientists and great thinkers by the time they leave here. So if we could just have one more round of applause for all of those people who make it happen. Also a shout out to Megan McCoy, our teaching fellow. And the long list of PhD students and postdocs that she recruited to help teach in the science course, who we don't have enough time to go through all of them. Also, I want to thank Narina from the Stem Cell Core and David and Esteban, David in the Imaging Core and Esteban recently, formerly of the Imaging Core, for giving some really great boot camp in growing cells and taking pictures of them. That actually made all the lab mentors' job easier. And thanks to Mosaic College Counseling for working with the students, and who will continue to work with the students throughout the year until they get their college decisions and decide where to go. It's every year, so we've had over 270 students in this program over the last 20 years, and every single one of them is matriculated at a 4-year college or university. Many of them at schools that I wouldn't be able to get into if I applied to them. So they clearly are doing a great job. But that said, the ones who really have done a great job this summer are our 24 fantastic interns. This day every year is always a mixed blessing because it's a fantastic opportunity to celebrate their accomplishments, but it's also very sad because they're all going to go back to school, and the energy level in all the laboratories is going to drop like a rock. It's, you know, they really bring a lot to this, and you know, I think it's important to emphasize that you guys did this. Okay, you did this. We facilitated it, but you did it. So that it's like, I'm like, I don't know how to finish the sentence other than to stop there. Yeah. And so, you know, and we've had I think we've had a lot of fun with you this summer, and I hope you've had fun. Don't forget that once you're in LA HIP, you're always in LA HIP. You know, keep us posted on how things are going. Contact us if you need help finding opportunities to stay engaged in research as your career goes on. And I just wish all of you the best, both for your presentations today and for your ongoing and growing academic careers. Speaking of which, we're going to hear from one of our former students who I'm particularly proud of because she was in LA HIP, and then she came back in the summer, and then she came back another summer, and she came back another summer. And now she's a first-year PhD student at USC. Brittany Hansen.
Hi, everyone. Is this the right mic? Okay, I'll use it. Thank you, Dr. Fry. There we go. So my name's Brittany Hansted, and I was in the class of 2019. I was obviously in Dr. Mark Fry's lab under Dr. Schumacher's mentorship, as well as Edie and John, who were PhD students then, and I think are still students now. So here's a little bit about me. I'm going to give you a small presentation of my first day of LA HIP and now to where I am. I'm currently in Dr. Merrill lab at the USC Keck School of Medicine. I am studying craniofacial development. And I would like to first off start by thanking everyone in Dr. Fry's lab and everyone who has ever helped with LA HIP. We had someone named Kira when I was around, and I know she is somewhere getting a higher degree education. So this is our cohort. There were only 8 students back in 2019. There was only so much funding for us. My hometown is here, Los Angeles, California, and I went to the Girls Academic Leadership Academy, or GALA, if any of you know that. Yay! I started LA HIP in the summer of 2019 in Mark Fry's lab, and I did work in the epithelium and the cell differentiation during IBD. It was much more detailed back then, but I don't remember much of it now. So obviously, as Mark said, I continued every single summer between the pandemic and college. So I continued through 2021 and 2023 through the program Step Up. And every single year, I was able to go back out to the NIH in DC and present my research. So here I am in college. So I went to Wellesley College out in Massachusetts, 30 minutes from Boston, and my major was biology with a minor in Latino and Latino studies. I did a lot actually, but I don't want to say all of that. I was fortunately enough, my school was offered the McNair program, so I was a McNair scholar, and that helps. Well, I'm not sure if it now helps underrepresented students in science, but that's what it did. And I was also nominated into Sigma Psi, which is a research honors society, which is really, really fun. And this is me and my college roommate, my sophomore roommate, being inducted into that honor society. And I did research in a plant lab, actually, under Dr. Kay Peterman, and I also did cancer and aging research with Dr. Richard Young at MIT. Through these years and through LA HIP as well as Step Up, I was able to go to Abercrombs. So that's me with that poster over at that conference, and I was able to do it all through college. And at the end, my final year, my senior year, I was able to get the Outstanding Presentation Award. So that was really fun. I was also part of the rowing team, which if anyone knows anything about that, it's waking up at 4 in the morning and going to bed at 8 p.m. during college, 6 days a week. So now that I'm in graduate school, I am finally in Dr. Merrill Lab doing craniofacial development. I'm an incoming second year, which is a big year if you know anything about the doctorate program. And I had a big decision between 2 graduate schools, and the one that I made is obviously to stay here at USC. It kind of felt right that I started my training here at LA HIP, and it felt like I should come back to USC to start my doctorate and all of that training. So LA HIP gave me a head start with research and it helped me realize that I actually enjoyed that research. It was kind of a stern pep talk that Dr. Bogetman gave me during boot camp that made me realize that. It led me to choose the PhD route instead of being an MD or an MD PhD. And the basic wet lab training that I was able to receive with LA HIP gave me a competitive advantage when I was applying to labs. Although I was rejected once for having too much experience, I was able to join a different lab in my undergrad that let me work straight in through the beginning of the academic year, instead of having to learn all technical skills from zero. Even small things like keeping a lab notebook or learning how to read scientific papers were a huge boost between undergrad and graduate school and high school. Being able to come up with ideas is something that I was able to practice over the years by doing research itself, like you guys are doing now. I'm so thankful to my mentor in Dr. Fry's lab, as well as Dr. Fry lab, as well as Dr. Fry. Dr. Schumacher, he really helped me become independent as time went on in the lab every summer that I kept coming back. I also really enjoyed the community and cohort that LA HIP was able to give me. Even though it was a very small group, it felt very tight-knit, and I feel like I have friends for a long time now. Training at CHLA through LA HIP to now becoming a PhD student at USC is kind of really full circle for me, and it makes me really appreciate this program and even more, and I hope that all of you students are able to do as much as I was able to do it and even more. And yeah, that is a little bit about me.
Thank you so much, Brittany. It's always wonderful. Oh, hold on, I like this one. I'm Danny. I am the LA HIP program administrator. Sorry, I totally didn't finish my thought, but thank you so much, Brittany. It's wonderful hearing students' experiences and the growth that they have, after the program. And once you're in LA HIP, you are in LA HIP for life. So expect me and Emmanuel to be emailing you. But also, I think it has been such a pleasure and a privilege to just watch how they have been in the lab, becoming researchers, and even more than that, like just the conversations that we've had for the past 8 weeks have been exceptional. And I told them this at the very beginning, that this was not an accident. And I think going into your senior year and beyond, I just want you to remember that you were selected to be in this program because you were special, and we saw that, and these presentations are going to exemplify that in so many ways. So parents, thank you all so much too. We know how hard it is to get everyone here by 9, hopefully 9. But we really appreciate you, and I mean, you know them as well as we do, and you've gotten to love them for probably longer, but we are so excited for today, and I am going to welcome up our first presenter, who is Mila from Culver City High School. Mila enjoys to do water polo and swim outside of the lab, and she enjoyed research experience and flourishing the nerdy community in LA HIP. Because of LA HIP, she looks forward to doing research in the future, and she said to choose one, but I definitely read both of them. So without further ado, let's welcome up Mila.
Hi, my name is Mila Stradaboom from Culver City High School, and I'm going to be talking to you about my work in the DIAS lab this summer regarding zona incerta inhibition and its impact on nucleus reunion activity. So post-traumatic stress disorder, or PTSD, is a mental disorder of persistent mental stress that impacts approximately 24 million people in the US at any given time. PTSD is characterized by a deficit in extinction, learning, and recall, which can be best explained through the following example. So there are some things that can be completely harmless, like California Girls by Katy Perry. But what about California Girls by Katy Perry playing in a bad car crash? After this incident, a person may exhibit a fear response to this song every time they hear it, like their brain thinks this is a cue for their body to prepare for something bad to happen. If they continue showing this fear to the song when played in the same context over and over again, that would be a deficit in extinction, learning, and recall, because they can't learn and remember that the song is not predictive of a harmful event. So we wanted to better understand the deficits in extinction, learning, and recall. So we started by looking at the zona incerta, or the ZI, a brain region that the DIAS lab is particularly interested in and has been implicated in the process of extinction. Given that brain regions are highly interconnected, we wanted to understand how inhibition of ZI neurons may impact downstream regions. Since the ZI has an extensive network of projections, we focused in on the nucleus reunion, or RE, which is important in fear and extinction, learning, and recall. So our question became, how does inhibiting ZI neurons impact neuronal activity in the RE? Given that the ZI is primarily made of inhibitory neurons, which make subsequent neurons quieter, we hypothesized that inhibiting these ZI neurons, effectively canceling out their inhibitory effect, will lead to more excitation in the RE. In essence, decreased ZI activity will lead to increased RE activity. So, to answer this question, my mentor modeled extinction, learning, and recall with mice after injecting a virus into ZI to inhibit cell activity during extinction recall. After, we sectioned these brains, cutting thin slices of tissues to subsequently label cells active at extinction recall in the RE using immunohistochemistry, and then we looked at these labeled cells in the microscope. So for our analysis, we first confirmed the expression of cellular activity in the RE, which is indicated by cells labeled in red from immunohistochemistry. We then calculated the total number of cells active at extinction recall bilaterally in the RE, comparing our experimental and control groups. We compiled our analysis in this graph, which essentially displays the average number of cells active at extinction recall in RE for each group, with the control groups in the light blue and the ZI inhibition group in the dark blue. We found that overall there is no statistically significant difference between our experimental and control groups. However, there is a trend of less cellular activity in the ZI inhibition groups versus the control groups, which is actually opposite of our initial prediction. So, recalling our initial question, does inhibiting ZI neurons impact neuronal activity in the RE? We concluded from our data that ZI inhibition has no impact on RE activity. Since the data is not significant, but shows an overall trend, we think that more data should be collected to confirm any differences that may indicate a relationship between ZI activity and downstream RE activity. We may also need to look at this relationship in a different way. For example, it would be interesting to see if shutting down ZI engram neurons could result in any shift in the RE or other downstream regions of the ZI. Thank you so much.
Wonderful. Thank you, Mila. Up next, we have Venice and Caitlin. Venice is from Granada Hills Charter High School, and Caitlin is from Synergy Quantum Academy. Their fun fact is that they hand sculpted a model of a kidney podocyte named Poot. Come on up.
Hello everyone, my name is Caitlin.
My name is Venice, and our presentation is silencing of AKT2 in podocytes modulate their cell cycle. So over 35.5 million people in the US face the challenges of chronic kidney disease, or CKD, where the kidney is unable to properly filter between the body's nutrients and waste. The kidney itself is composed of nephrons, which have a glomerulus at the end where majority of the filtration occurs. The glomerulus contains a filtration barrier, as you can see in the top image, or a model that's composed of 3 different layers that help prevent large protein molecules in the blood from escaping through the urine. The bumpy blue layer at the top are podocytes, or visceral epithelial cells, and these are attached to the outer edge of the membrane and interact through different foot processes crucial to filtration. So zoning into a podocyte itself, as you can see in the bottom image, that podocytes are associated with different signaling pathways that communicate whether or not it should enter the cell cycle. So our research specifically looks at a pathway that contains a protein called AKT2 that we hope to silence by using siRNA to block the translation of mRNA molecules. And we also looked at another protein marker called nephrin, which indicates healthy podocyte characteristics by helping maintain its foot processes.
And a little background on the cell cycle in podocytes. Podocytes stay in G0, the resting phase, and this phase is regulated by P21, which is a highly expressive protein. Normally, cells would enter the cell cycle and they would proliferate. However, podocytes only do so when damaged because they are highly specialized cells. Proliferation or cell division is bad for the podocytes because it forces them to detach from the glomerulus, which results in loss of characteristics and essentially poor filtration in the kidney. So it is important for them to remain in G0 and that P21 is expressed. And for our research question, what is the role of AKT2 in regulating the podocyte foot process and cell cycle? And we hypothesized that silencing AKT2 with siRNA will contribute to the change in the podocyte's expression of nephrin and the cell cycle regulator P21. For methods, we began our cell culture by differentiating kidney progenitor cells derived from amniotic fluid into mature podocytes. And once matured, we split the cells and we categorized them by our control and our different time points. To silence AKT2, we added siRNA and we collected their sample at their given time. And the reason why we do this is because we want to know at what specific time period the siRNA plays its role at interfering with the AKT2 expression.
We then performed a Western blot to confirm that the siRNA was actually effective by using antibodies to detect the presence of AKT2 over time. We also performed one to target P21, a cell cycle regulator protein. And after having that baseline test, we were able to use immunofluorescence microscopy on our podocyte chamber slides to show the effect of silencing AKT2 on nephrin expression. So now looking at our Western blot results, we see that the total, from the first row, the total amount of AKT2 present in the podocytes over time, and from this we can confirm that the silencing did in fact work as the band intensity starts to fade over time. And we also used vinculin as our housekeeping or control because it's a protein present in all cells, and the homogeneous band intensity throughout shows that only the AKT2 was silenced by the siRNA and other proteins weren't affected.
Our immunofluorescence imaging determines if the podocytes maintain their healthy state or not. And as we can see, depending on the visible nephrin, and as we can see, the images show the staining of the nucleus in blue and the nephrin in green. And nephrin is basically the main marker of the podocytes. It is expressed when they are in their healthy state. And when we added the siRNA, we can see that over time there is a significant loss of nephrin, and this just proves that the silencing actually caused the podocytes to lose their characteristics and their functional state.
So now going back to the connection between AKT2 and the cell cycle, looking primarily at the P21 expression at the first 3 rows with the control group and the last 3 rows at the 48-hour mark where silencing is most effective, we see that there's a reduction in abundance, indicating that P21 is in fact affected and that reducing AKT2 can potentially induce podocytes to re-enter the cell cycle.
And to conclude, AKT2 plays an important role in podocytes because it regulates nephrin and P21. And for our potential future work is to add trypan blue staining to show the ratio of the dead cells in the podocytes. Signs for AKT2, we will confirm that silencing AKT2 affects the cell cycle by modulating its cell cycle regulators, and further test this hypothesis of AKT2 expression in human biopsies from different chronic kidney diseases. Thank you all, and a special thank you to our mentors.
Amazing. Thank you so much, Caitlin and Venice. Now I'd like to welcome up Natalie and Brandy. Natalie is from Mace's. She likes to try new food and has enjoyed learning about retina cells. Brandy's from Gardena High School, and he loved the lab experience and being able to meet people who share similar experiences to himself. So without further ado, I'm going to welcome up Natalie and Brandy.
Hi everyone, my name is Natalie.
My name is Brandy.
And today we're going to be presenting our DAPI-based ultrastructure expansion microscopy in human retinal organoids, a pilot study.
So from some background, we can't clearly see tiny retinal structures with standard or confocal microscopes because they don't zoom in enough and have a lack of resolution. For example, photoreceptor cells in the retina, which handle night, day, and color vision, contain a protein transport hub called the photoreceptor sensory cilium. In diagrams, it looks simple, but when we actually try imaging these for research purposes, it's a blurry mess. The only reliable method has been electron microscopy, but that itself is expensive, slow, and requires specialized training. Specifically in the Nagel lab now, we're interested in where proteins are located in the retina, but we have the same visibility issue. So we ask, can ultrastructure expansion microscopy enhance the spatial resolution of synaptic protein localization in the outer plexiform layer of the retina?
Okay, so ultrastructure expansion microscopy, or UEX for short, is a technique that physically expands tissue, allowing researchers to see nanoscale structures using the standard confocal microscopes to help see retina cells like photoreceptors, which helps detect light. In this image, you can see that the right image with the no expansion tends to be less defined than the one with UEXM. So in Dr. Nagel's lab, they are currently studying human retinal organoids, focusing on proteins in the OPL where photoreceptors connect with bipolar cells, a messenger cell. Key proteins like TRPM1 appear blurry under confocal microscopy, even at maximum zoom, making quantification difficult. Therefore, we hypothesize that by using UEXM, we're able to see the snapping of synaptic proteins in the OPL.
So we followed a 5-day protocol to try out this method. On day 1, we had organoids that were fixed to preserve the structure and anchored overnight in a formaldehyde acrylamide mix. On day 2, a gel had formed around the organoids with a monomer solution. We then added the organoids into a denaturation buffer with cover slips added to improve antibody access for later on. Days 3 and 4, the gel had expanded significantly, but we can't use all the gel, so we trimmed it. And using what was left over, we incubated it with primary antibodies to target specific proteins, and then fluorescent secondary antibodies, and then stained with DAPI to label the nuclei. And on the last day, we coated chamber slides with poly-D-lysine to prevent gel drifting during imaging, transferred the gels, and prepared them for the actual imaging.
Okay, so some results that we got is, in this image, you can see a petri dish with an organoid and the gel, and it's about 1 centimeter long pre-expansion. Post-expansion, the gel group's about 5.5 centimeters. So for our research, we had 6 samples, and we took the total width of expansion and divided it by the 6 samples we had, and on average, there was a 4-time increase of the gel. Our results came out so beautiful. Right here on the left is the no expansion, and to the right we have the UEXM. So As you can see, normally that at 10x, we can't see the OPL layer, even at 63 times magnification, which the proteins are still hard to distinguish. But with UEXM, we're able to see the OPL much clearer. So you can see it right here, whereas with the no expansion, it tends to be harder.
So, based on the results, they show that we did successfully expand the organoids, which did improve the retinal layer visibility during imaging and clarified the OPL, which is our lab's main focus. However, some proteins didn't emit signals, which is a key limitation as of now. But looking forward, UXM has strong potential. If we're able to optimize the antibody and staining protocols, which could help localize protein more reliably, that would give us exactly what we need for the lab in our research. With refinement, UXM could transform how we study cell connections and protein localization beyond just the retina without having to rely on just electron microscopy. Special thanks to everyone in the Nagel Lab, including Dr. Nagel, our amazing mentor, Layla, and Patty, and everyone else who helped with the project.
Thank you.
Oh, sorry. Thank you so much, Natalie and Brandy. Up next, I would like to welcome up Esteban and Alan. Esteban is from Francisco Bravo Medical Magnet, and Alan is from Azusa High School. Together, they both enjoyed connecting over the summer with people with similar backgrounds and looking forward to the growth of their common love of STEM. Come on up.
Good afternoon, everyone. My name is Esteban.
My name is Alan Ramirez.
And we're going to be presenting our research titled PAX3-FOXO1 Fusion Drives Cell Cycle Progression Dysregulation in Fusion-Positive Rhabdomyosarcoma.
To begin, we're going to talk about a little background about our rhabdomyosarcoma. Rhabdomyosarcoma is the most common pediatric soft tissue sarcoma, and there is two main subtypes, which are alveolar and embryonic. We won't go deeply into these two subtypes because it wouldn't be enough time. And alveolar RMS is basically just the fusion positive gene of PAX3-FOXO1 fusion gene, which is just two separate genes that fuse together. And we call this basically fusion positive rhabdomyosarcoma, or FPRMS for short, and it is more worse outcomes than embryonic, causing more aggressive form of cancer.
So what is PAX3-FOXO1 fusion gene? First, PAX3-FOXO1 is a fusion gene, meaning it forms when the PAX3 gene found here on chromosome 2 fuses with the FOXO1 gene found on chromosome 13. This abnormal combination creates a new hybrid gene with serious cancer-promoting properties. PAX3-FOXO1 is also oncogenic, meaning it plays a role in tumor initiation and promotes cell proliferation. Throughout dysregulation of the cell cycle, it can be included that PAX3-FOXO1 is important in rhabdomyosarcoma. It's also important to point out that PAX3-FOXO1 does not cause rhabdomyosarcoma by itself, hence the subtype FPRMS. Instead, it contributes to multiple factors that lead to cancer, acting alongside other genetic and molecular changes throughout the cell.
So about the cell cycle, it is really important for our research and also in everyday life. And this is because in the cell cycle, cells grow, copy their DNA, and divide into different other cells. And basically, if we think about it, a 9-year-old turns into a 12-year-old body, basically growing, once again, a cell cycle. And also, if that 12-year-old gets a boo-boo, it has to repair itself. So cell cycle once again. And controlling this cell cycle is CDK activity, which is cyclin-dependent kinases, which they control the timing of the cell cycle and make sure everything's going great. And uncontrolled cell cycle or uncontrolled CDK activity can cause cancer.
So here was our research question in our lab. How does the PAX3-FOXO1 dysregulate the cell cycle?
To figure out this, we used a method called immunofluorescence staining, and where we compared phosphorylated CDK2 activity and gamma H2AX expression. In phosphorylated CDK2 activity, it is a marker that helps basically says that the cell is growing and dividing, and it's found in the nucleus where the cell is actively dividing. And gamma H2A marker is basically also a marker that shows DNA damage, and it's saying that the DNA is broken. It signals to the cell that there's DNA damage, and there appears as spots on the nucleus.
So here are our results. Immunofluorescence shows that PAX3-FOXO1 knockdown, or PFKD, has lower pCDK2 concentrations. This less bright red cells means that during the cell cycle, accurate progression through checkpoints isn't occurring. PAX3-FOXO1 knockdown also has higher gamma H2AX concentrations. As you can see, the brighter green dots in the middle of the cell indicates that there is more DNA damage occurring within the cell's nucleus.
Also, in research, we would want to have our results broad and diversified. So we use RH30, which is another cell line, and we've seen similar results that were in the first cell line, which was RH41, and it basically supports the generability of the findings.
Besides the immunofluorescence, we also wanted to reinforce our findings with a Western blot with 2 markers. Phospho-CDK2, as noted here, is lighter in PAX3-FOXO knockdown cells, which is suggesting arrest, while phosphohistone 3 is also lighter in the PAX3-FOXO knockdown cells, suggesting decreased proliferation occurring in these RMS cells.
For our conclusions and future works, we have identified that PAX3-FOXO1 drives oncogenesis by altering cell cycle and possibly increasing replication stress. Decreased phosphorylated CDK2 activity and elevated gamma H2AX support disrupted cell cycle and DNA damage.
Since our lab identified certain downstream targets suppressed by the PAX3-FOXO1 fusion protein, a type of transcription factor, our data highlights inhibition of these proteins that offer a potential therapeutic strategy in fusion-positive rhabdomyosarcoma. Given that transcription factors like PAX3-FOXO1 are notoriously difficult to target with drugs, protein-targeted therapies present a much more viable and actionable target for the development of novel therapies in rhabdomyosarcoma. We want to give a big thanks to the Jinsuk Park lab at CHLA, our wonderful PI, Jinsuk Park, and our amazing mentor, Dr. Chanda Vemula, for all of his help. Thank you.
Thank you. Now I would like to welcome up Flo and Jessica. Flo goes to West Ranch High School, and this summer enjoyed meeting people and loved the people and the mentor, and looks forward to having many cats in the future. Jessica goes to University High School and enjoyed applying concepts that she had only read in textbooks before, and looks forward to returning in the lab this summer. Or winter. Oh my gosh, this winter, yes. Please come on up.
Hi everyone, my name is Jessica.
And I'm Flo, and we're from the Dr. Kami Takahara's lab.
And we'll be presenting on the genetic differences underlying differential responses to semaglutide. Obesity is one of the biggest public health crises in America today. 1 in 3 Americans have obesity, and you should be concerned about this because obesity leads to increased risk for heart disease, stroke, diabetes, and a plethora of other conditions.
And so semaglutide, which is a GLP-1 receptor agonist or activator, has emerged as an effective treatment for obesity, so as it helps manage blood glucose levels, regulate appetite, and promote weight loss, just like the hormone GLP-1. And 1 in 8 Americans have actually taken semaglutide, which you might be familiar with in the form of Ozempic or Wegovy.
So amidst the growing popularity of semaglutide, we've begun to see a variety of responses to the drug. In a study of 7,000 people, most people saw average or significant weight loss. However, there's a small percentage, 10 to 15% of people who either don't lose weight or even gain weight on the drug. And our research is asking why these different people have different responses to semaglutide.
And since we're a neuroscience lab, we're going to focus on the brain. And we hypothesize that these differential responses to semaglutide are due to generic differences in brain regions that respond to the drug, specifically the arcuate nucleus of the hypothalamus, or the ARC, and the nucleus of the solitary tract, or the NCS. And the way that semaglutide works in the brain is that it's going to be first injected into the body, and that's going to make its way through the bloodstream all the way to the brain. It's going to reach the nucleus of the solitary tract. and that region is involved in satiety. And the drug is also going to interact with the arcuate nucleus, a region of the brain that creates adaptive responses to weight loss. And to model this, we're going to use 2 strains of mice to model that just a genetic diversity of these responses between people. We're using New Zealand obese mice, NCO mice, which lose little to no weight and even gain weight, just like the 10 to 15% of users we saw, which you can see in this graph. They didn't lose much weight. And that's compared to the C57 mice, which do lose a significant amount of weight, like the other users.
For our experiments, we started off by giving these 2 strains of mice a high-fat diet until they gained 50% of their weight. They were then put on a 3-week semaglutide treatment. After that, we dissected their brains, took brain punches specifically of the ARC and the NTS, and isolated RNA from those samples for bulk RNA sequencing. Using the sequencing results, we were able to do data analysis on these samples.
And so we generated this volcano plot, and this is going to show which genes are differentially expressed between the NCO and the C57 mice. That's going to show changes in transcription of the different genes to RNA. So the changes, basically changes in the number of instructions. And so anything downregulated will be to the left and in blue, and anything upregulated will be to the right and in yellow. So we basically see a downregulation of genes encoding for ribosomal proteins, and ribosomes are the things in the cells that produce proteins. We also saw an upregulation of WDFY1, a gene involved in inflammation. And we saw no change in GLP-1 receptor transcription levels, which is interesting because that's where the drug binds to. And we see pretty much the same thing in the NTS, downregulation of genes encoding for ribosomal proteins, upregulation of genes involved in inflammation, and no change in GLP-1 receptor transcription levels.
On to our pathway analysis. So in the ARC, we see an upregulation of inflammatory pathways, meaning that in the NZO mice, there's more inflammation in comparison to their C57 counterparts. And at the same time, we see a downregulation in translation pathways, meaning that these NZO mice are essentially making less functional proteins than their C57 counterparts. A similar story is retold in the NTS, where we see an upregulation of inflammation and specifically neuroinflammation, as well as a downregulation in translation.
And so to sum it all up, GLP-1 receptor transcription levels, or the instructions made, are not significantly different between the NCO and the C57 mice, but we do see an upregulation of inflammatory pathways, which indicates that the drug isn't working neither for weight loss nor to reduce inflammation. And we also see a downregulation of translation pathways and ribosomal proteins, so fewer components of the cell's protein building machinery are being made. So that may cause the cell to make fewer proteins overall, including GLP-1 receptors.
So future investigations could measure GLP-1 receptor expression in these NZOs versus C57s to test if the changes from semaglutide lead to decreased expression of the product and not the instructions as we tested in this experiment.
And a special thank you to our beautiful lab over there.
Thank you. Now I would like to welcome up to the podium Sina. She's from Harbor Teacher Prep Academy.
Hi, my name is Sina Fesahaya, and I'm working in the DIAS lab, and I'm focusing on the downstream impact of zona incerta inhibition on ventrolateral periaqueductal gray, or the VLPAG. So post-traumatic stress disorder, or PTSD, is a condition of lasting mental and emotional stress. Affecting approximately 6.8% of Americans, PTSD is highly pervasive and as such of great importance to understand. PTSD is characterized by deficits in fear, learning, and recall, so we aim to better understand these deficits. To accomplish our aim, we looked at the zona incerta, or the ZI, which is a brain region involved in learning and memory that the DIAS lab is particularly interested in. Given that brain regions do not act in isolation but are highly interconnected, we were curious if the manipulation of the ZI within the process of learning and memory might impact downstream regions. We focus on the projection from the ZI to the ventrolateral periaqueductal gray, or the VLPAG, given that they are both implicated in fear behavior. So we asked, does inhibiting these ZI neurons influence neuronal activity in the VLPAG? So before crafting our hypothesis, we had to consider the composition of the ZI. The ZI is predominantly made up of GABAergic neurons, which are inhibitory. And inhibition works like a car brake that slows a car down. As such, GABAergic neurons effectively slow down activity of neighboring neurons. Since we want to inhibit ZI neurons, which are primarily inhibitory, it is like we are removing the car brakes that are the ZI neurons. So we hypothesized that inhibiting ZI neurons should lead to downstream VLPAG neurons to become more excited. To investigate our hypothesis, our mentor first injected a virus into the ZI of the mice to inhibit these neurons during behavior tests, which model fear, learning, and memory. After completing behavior, the brains of the mice were dissected and sectioned to prep them for labeling and imaging. Then we used immunohistochemistry to label cells active in the VLPAG. And finally, we used microscopy to analyze cell activity in the VLPAG following ZI inhibition. This is an example of a brain section that we examined. The red points are indicative of cells that were active in the VLPAG. We counted the number of cells to assess the level of activity between our experimental and our control groups. Looking at the number of cells active as averaged across the size of the VLPAG, we see no statistical difference between control groups, which are in light blue, and our experimental group, which is in dark blue. We had initially asked if inhibiting ZI neurons influences neuronal activity in the VLPAG and hypothesized that disrupting this pathway would cause the VLPAG activity to increase. However, we do not see any difference in the VLPAG activity between the control groups and the ZI inhibition group. Thus, we conclude that inhibition of ZI activity does not have a downstream consequence on VLPAG activity. However, it would be interesting to see if doing the opposite or boosting ZI activity might have an effect on VLPAG activity or other downstream regions, and if this may improve learning and memory. So special thanks to our PI, Dr. Diaz, and our mentor, Haley. Thank you.
Thank you, Sina. Now I'd like to welcome up Lila and Isabel. Isabel's from Birmingham High School and Lila is from San Pedro High School. Isabel said that LA HIP allowed her to explore a career in research, which is something she hadn't thought she wanted to pursue prior. She loved learning about the different protocols and procedures done in the lab and she says she looks forward to returning to the Fry Lab and continuing to explore research in the future. Lila really loved working with her lab partner, and she also said that she liked to visit the cafeteria. In the future, she hopes to come back and work in a lab or one with a similar environment that she had here, and would love to work with her mentors again. So without further ado, Lila and Isabel.
Hi, my name is Isabel.
Hi, I'm Lila, and we're from the Fry Lab.
And we'll be investigating the role of latrofilin-2 in intestinal cell physiology and cancer. So colorectal cancer is one of the leading causes of cancer-related deaths in the U.S., affecting both men and women. And one of the most dangerous aspects of this disease is its ability to metastasize, meaning that the cancer can spread to other parts of the body, making it much harder to treat. The progression of colorectal cancer, as depicted by this graphic, begins with abnormal cell growth that can then progress into a malignant tumor. Cancer cells can then migrate into the extracellular matrix, which is just a network of proteins that provides tissue structure, invade blood vessels, and ultimately spread throughout the body. Our lab is investigating a G protein-coupled receptor called latrofilin-2, or ADGRL2. And this receptor sits on the surface of cells and helps the cells interact with their environment. Latrofilin-2 expression is frequently altered in cancers, and whether it's up or down depends on the type of cancer it is, which is why we're interested in its role in the colon. So on the left, we reanalyzed public gene expression data to focus specifically on latrofilin-2 levels, and in early stage colorectal tumors, also known as colorectal adenomas, latrofilin-2 expression is being lost, which could indicate its role as a potential tumor suppressor. On the right, the graph focuses on latrofilin-2 expression across cancer cell lines. And in colorectal cancer cells specifically, the expression is dramatically reduced compared to the other lines, which further supports the idea that the loss of this receptor could be contributing to cancer development or progression.
And so our research question is, what role does latrophilin-2 play in colonic epithelial cells? And with that, we hypothesize if latrophilin-2 is a tumor suppressor, then overexpressing it will decrease tumorigenic properties like metastasis. And so to address this, we use HCT116 human colorectal cancer cells and compared them to HCT116s that were altered overexpressing latrophilin-2, our gene of interest. And so to investigate the role of latrophilin, we conducted a transwell migration and invasion assay. The transwells are cups placed into a well plate in which divide the cell culture area into two chambers, the apical side, which is the cup, the top side, and the basolateral side, which is the bottom side, with the porous membrane in between. On the apical side, it holds the cells, and on the basolateral side, it holds the media. And so we tested migration by seeing how the cells cross the membrane to the basolateral side, and we tested invasion with the addition of a matridrel to model the extracellular matrix, resembling how cancer cells invade tissue. And so after plating, we incubated the cells to allow them to move from the apical side to the basolateral side. And then after the incubation period, we removed the cells that did not migrate from the apical side with a cotton swab, in which the cells that did migrate to the basolateral side were further stained with crystal violet for visibility for further imaging and counting. And so we tested two conditions, in which the first condition had starved media, DMEM, in which lacked cell nutrients, and then the second condition was complete media with 10% fetal bovine serum, in which promotes cell growth, which serves as a chemoattractant. And so our results show that cells overexpressing latrophilin-2 had reduced both migration and invasion compared to the parental HCT116 cells. And so the trend shows a lower number of cells passing to and through the membrane. However, we can also observe that cells overexpressing latrophilin-2 in the presence of a fetal bovine serum, fewer cells remain on the membrane overall. This could mean that more cells had migrated completely into the media and were counted, potentially underestimating total movement.
So to address this, the lab plans to refine the assay by also collecting and staining the cells from the bottom of the culture wells and not just those on the membrane, because this would give us a more accurate picture of total migration and invasion. In summary, our findings suggest that latrophilin-2 suppresses key steps in the process of colorectal metastasis, which makes it a potential therapeutic target for colorectal cancer.
And special thanks to the Fry Lab, and shout out to Team Latrophilin and John.
And Mudang. Up next, I'd like to welcome up Benjamin and Josiah. Benjamin goes to Mark Kreppel High School, and Josiah is also from Bravo Medical Magnet. This summer, Benjamin enjoyed working under the hood and working with cells and hopes to pursue a future career in STEM. For Josiah, he really enjoyed gaining hands-on research in the lab with passionate mentors who challenged him to think critically. In the future, he does plan to pursue biomedical science and pursue a career where he can see bridge the gap to cutting edge discoveries. Where are they? Oh, there they are. Up next.
Hi, my name is Benjamin Carvajal.
My name is Josiah Reforzo.
And our experiment is on the short and long-term effects of microplastics on glomerular kidney cells.
So what are microplastics? Microplastics are synthetic polymer particles less than 5 millimeters in size, often resulting from the degradation of larger plastic material. And these particles have been detected in human biological fluids, including blood, urine, and placental tissue. So what is the problem? The problem lies in the fact that as our blood gets filtered by our kidneys, they are exposed to small quantities of microplastics. And the impact of chronic microplastics exposure on these renal cell types remains poorly understood.
And so what are kidneys and what is the glomerulus? Inside of your body, there are two organs that help to filter your blood and remove any solutes or waste to later dispose of as urine. These are your kidneys. Inside of your kidneys, there are things called nephrons, which are just the smallest working unit inside of the kidney. And inside of the nephrons are your glomerulus. The glomerulus does the brunt of the work for the kidney by filtering your blood. And inside of the glomerulus, we wish to experiment on 3 different cell types: podocytes, glomerular endothelial cells, and mesangial cells. And our research question for this experiment was how does short and long-term exposure of microplastics affect glomerular endothelial cells, podocytes, and mesangial cells? And we hypothesized that there would be a significant decrease in functional protein expression, an increase in cellular stress, and significant changes to the morphology of the cells left alive or just their shape.
So for our methodology, we began our experiment by culturing glomerular endothelial cells, mesangial cells, and podocytes. And for the podocytes, we differentiated them from adult kidney progenitor cells derived from amniotic fluid. We then plated the cells onto 6-well plates and 8-well slides, treating them with polystyrene microplastics. We then waited for 3 and 15 days for our short and long-term exposure groups, after which we ran a Western blot to search for proteins that induce apoptosis or cell death. And using immunofluorescence, we stained the 3 and 15-day cells for protein markers that indicate cellular stress and other vital functions for the cells. And on this slide, we have our light microscopy photos of all 3 groups. On the top row, we have our control group, which were not exposed to microplastics. And on the bottom row, we have our experimental group, which were exposed to microplastics. And as you can see, there's a significant decrease in cell count across the glomerular endothelial cell and podocyte group. We also see a significant morphological change in the JEC group. In the control group, they are much more strand-like, and in the experimental group, they are much more granular and circular. And the mesangial cells saw like little to no change after introduction to microplastics.
So here are our immunofluorescence photos. In all of the pictures, the blue represents the nuclei of the cells and the red represents the microplastics. On the left, we tested for ROMA1, which is an indicator for oxidative stress in the cells. And as you can see, the cells are under more oxidative stress when under microplastic conditions. This effect was seen among all cell types. In the middle, we tested for CD31, which is a protein used for the cytoskeletal structure of the JEC cells. And as you can see, there's less CD31 in the microplastic group, further explaining why there were morphological changes among the microplastic group. On the right, we tested for podocin, which is a protein vital for the podocyte's role as a barrier for the glomerulus. And as you can see, there's less podocin for each cell among the microplastic group. This leads to them becoming smaller and more isolated in the experimental group, worsening their function as a barrier in the glomerulus. You can also see that there's differing amounts of microplastic intake within each cell, which with the podocytes taking in the most amount of microplastics and mesangial cells taking in the fewest.
And looking at our results from our Western blot, we can see a substantial increase in the concentration of BAX, which is a cell death inducing protein in the JEC experimental group compared to the JEC control group, explaining the decrease in cell count.
And so our conclusions for this lab were that there were increases in oxidative stress among all cell types, and that there was cell death and morphological changes experienced by both the podocyte and JEC groups, while mesangial groups remained largely unchanged. And all of the measured effects were amplified across the 15-day groups from the 3-day groups. And microplastics are a lifelong issue. So I'd want you to know that this is just 15 days. Imagine what would happen across your entire lifetime.
And some future work can include testing microplastic effects on glomerulus on a chip systems to emulate in vivo interactions between different cell types, and comparing effects of different plastic types and particle sizes on glomerular cells. In our experiment, we used polystyrene.
And special thanks to Stefano Dasaco, Mohamed Kasravi, Nancy Marzano, and Rachel Wen.
Those microplastics. Anyway, up next, I would like to welcome up Light from Lawndale High School. Her favorite thing to do outside of the lab is to sing and write poems. She said that over the summer, she enjoyed the people that she's met along the way, the lab work, and the food, and of course, the joy, laughter, and tears that came along the way. She looks forward to meeting them again, and the food, of course.
I'm a very tall one, you know. All right. So good afternoon, everyone. My name is Light Phillips. I'm from Lawndale High School, of course, as introduced. And today I will be presenting my research on the effect of maternal immune activation on astrocytic numbers in postnatal brain. All right, so maternal immune activation damages the fetal blood-brain barrier structure during pregnancy. This happens when severe flu activates inflammatory response, like you can see over here, which is when the immune system of the mother begins attacking the developing fetus, leading then to the permeability and leakage of the fetal blood-brain barrier, which is a structure that protects the integrity of the brain. Damages to the BBB at early stages can lead to chronic neuroinflammation, increasing the risk of neurological disorders such as schizophrenia later down in life. Now, star-shaped glial cells, which is what I'm focusing on, such as astrocytes, which develop postnatally or after birth, are important for maintaining offspring BBB. However, their role in BBB disruption following MIA remains unclear. By hoping to clear up a little part of this mystery, we ask ourselves an important question. Does MIA, maternal immune activation, affect astrocytic contribution to the blood-brain barrier in the cortex and corpus callosum in PD30 offspring adolescent brain? So my hypothesis is that MIA may alter astrocyte numbers in adolescent offspring, potentially contributing to long-lasting effects and changes in BBB development and functions. For our methodology, my mentor, Hannah, injected poly-IC, which mimics maternal inflammation in a pregnant mice, and saline, our control, here in a different pregnant mice. And then, once their offsprings were born at adolescent age PD30, their brains were extracted and then sectioned. After that, immunohistochemistry, also referred to as IHC, was performed on them, which is a process that stains for astrocytes and other compounds of the brain using specific antibody markers, which is later then visualized in a microscope here through fluorescence lighting and analyzed through image J. Right. So once we analyzed this, we saw a trend, a decrease in poly-IC astrocytes that were treated with poly-IC. As you can see closely, hopefully, that their astrocytes are abundant in the saline group, which fetal development. And however, in poly-IC, you can see that the astrocytes are either reduced or they have some, changes to their morphology. Here, if you can, yeah, perfect, you can see that, great. So here, the astrocytes, the process of the branch is thicker, and this shows that it's trying to compensate for the damages to the BBB, and this can have long-term consequences because this can mean neural damage or even cell death, right? And then here in the corpus callosum, you can see this is a very pretty picture, and saline that the astrocytes are abundant and they're doing what they're supposed to do. However, in poly-IC, they are reduced, and you can see that there is difference in the cell numbers. Yeah, perfect. So for our conclusion on our future works, so we know that MIA decreases astrocyte cell numbers in the corpus callosum and primary motor cortex. Astrocytes are key to maintaining BBB integrity and protecting the developing brain. Their loss or dysfunction amplifies susceptibility to cognitive and psychiatric conditions. Further research should explore interventions that restores astrocyte functions or strengthen BBB integrity in high-risk pregnancies, hopefully paving the way for neuroprotective treatment. And before I leave, I would love to leave you guys with a simple reminder that one small step into research is one giant leap for mankind. It was inspired by Neil Armstrong. Thank you.
Thank you, Light. Up next, I would love to welcome up Miriam and Lucero. Lucero is from USC Hybrid High, and Miriam is from North High School in Torrance. They both enjoyed meeting new people and having the opportunity to make connections in the lab, and they loved each other as lab partners, which is so cute. Without further ado, I'd love to welcome up Miriam and Lucero.
Hello, everyone. Thank you for joining us today, whether you're here right now or on virtually. My name is Miriam.
And my name is Lucero.
And we are investigating the impact of early life high-fat diet on vagal circuits. To begin, obesity is a growing problem in the United States. We have 1 in 5 children and 1 in 3 adults in the U.S. who are obese. And this is important because obesity can result in many health complications, such as cardiovascular disease, diabetes, certain types of cancers, and much more.
So what do we know about an early high-fat diet and cholecystokinin during development? We know that when you eat a meal, nutrient-sensitive cells in the gut release a hormone called cholecystokinin, or CCK. CCK then attaches to receptors on the vagus nerve, which then activates neurons in the nucleus of solitary tract, or the NCS, in the brainstem to tell us when you're hungry. So then our lab wanted to further understand how an early high-fat diet affects how mice feel full after eating while being controlled by the CCK. And they tested this by injecting CCK, and to measure the response to the injection, they They fasted the mice overnight, and they saw that in the control mice group at postnatal 35, which is early adolescence, the mice ate inconsistently after the CCK injection, which means that their fullness circuit wasn't fully developed yet. On the other hand, at the same time period, they saw that the early high-fat diet mice acted like more mature mice, ate less, and had a stronger satiety level response to the CCK injection.
To go further with our research, the region that we're particularly interested in is called the nucleus of the solitary tract, also known as the NTS. And this region of the brain, outlined in yellow, is responsible for feeding behaviors and satiation, which is fullness. And specifically in this case, we are looking for changes in activity in the NTS in response to that CCK injection, which you can see with all this bright red signaling within the yellow. That's all the CFOS, which is a protein marker for that CCK being signaled. And that brings us to our research question, which is what is driving differential responses to CCK? And we have a couple of hypotheses, one of them being differences in neuronal activation between the early life high-fat diet mice and the control mice are what are driving differential responses to CCK.
And our second hypothesis being differences in synaptic density between the early high-fat diet and control mice are driving differential responses to the CCK.
To test our hypotheses, we began with the pups of C57 mice split into two groups. The first group at the top was given a high-fat diet from postnatal day 0, also known as P0, to postnatal day 21, and the other group was given a standard chow diet. This was up until P21, which they were weaned away from their mother and both transitioned to the standard chow diet, up until P34 when they were fasted overnight before receiving an injection of CCK on P35. We waited an hour and a half because that's when CFOS, that protein marker, peaks, and then we perfused the mice with PBS to wash the tissues and PFA to fix the tissues.
And after our tissue collection, we froze our tissue in OCT. We then cryo-sectioned for our region of study, which is the NTS. Following, we used immunofluorescence to stain for neurotrace blue, which is an anatomical marker to help us visualize our region of study. We also stained for synaptophysin, which is a protein found in presynaptic vessels and in neuroendocrine cells. We lastly stained for CFOS, which is a protein marker for a specific stimulus, so in this case being the CCK. And we finally used microscopy to image and to help us quantify our CFOS and our synaptic density.
Upon quantifying the CFOS, these are the results. We do not see a significance in the data between the experimental groups within a sex, although there is a trend toward increased activation within the male mice, and this tells us that it's likely not the neuronal activation that is responsible for the differences in response to CCK. However, there are larger differences in the data when looking between the sexes, specifically with the male chow group and the female chow group. This is a similar finding to something we saw in our preliminary model, and finding out exactly why that difference exists could be something to investigate further.
And for our results for synaptophysin, we found the percentage of synaptic density by measuring the area fraction of our images, and we see no significant difference in synaptophysin in males. However, there is a trend toward decreased synaptic density in the females, and this tells us that changes in synaptic density are probably not responsible for the responses in response to the CCK, but future work should allow us to follow up on the small effect that we see in the females.
To conclude, we observed with our data that differences in neuronal activation in the NTS between the mice groups is not what is driving the differential responses to CCK. A potential future work that the lab could do would be to count the CFOS in the nodus ganglion, which is located right here in the vagus nerve, rather than the NTS, because that is the first node of the circuit between the gut to the brain.
And for conclusions for synaptophysin, we see that differences in synaptic density in the NTS between the mice groups is not what is driving differential responses to the CCK, and potential future work would be to use a different marker for synapses and or check different ages in the mice groups.
And that is all. Special thank you to Kamitakahara Lab and Levitt Lab, and thank you, Anna, who is on virtually.
Thank you. up next we have Nola and Milan. Nola is from North Hollywood High School and Milan is from Walnut High. Nola said, I loved LA HIP because of the community that we formed that led to lifelong friends. Love you, Milan. I look forward to pursuing biology in college and continuing to learn the hidden treasures in this field. Milan said, what I enjoyed most about LA HIP was the new connections and a lot of new friends. Love you, Nola. I look forward to going to college and studying biology. Welcome up. Come up here.
Hi everyone, my name is Nola.
I'm Milan.
And this is our presentation about targeting TBO1 and T-ALL.
So normal T cells are a specific type of white blood cell that play a crucial role in our immune system because they help the body fight infections. But in T-cell acute lymphoblastic leukemia, or T-ALL, these normal T cell functions are lost. T-ALL is a fast-growing cancer where immature T cells become cancerous and crowd out healthy blood cells in the bone marrow. And the bone marrow is where all blood cells are produced. So T-ALL is an extremely common type of blood cancer diagnosed in children. And there have been increased therapies like chemotherapy, as you can see from the survival curve, that have helped the rates of survival over time, but there is still a 20% relapse rate.
So TBO1 is a new target in Wnt signaling. Now what Wnt signaling does is control normal blood stem cell development, and so it's overactive in many leukemic stem cells, allowing them to grow and spread pretty quickly and easily. So targeting Wnt signaling has not given ideal results, but we hypothesize that TBO1, this new target, can provide better and more accurate information results.
So our research question is, does downregulating TBO1 affect the viability of leukemia cells and potentially reduce chances of relapse in T-ALL patients? And our hypothesis is that TBO1's role in Wnt signaling is crucial for T-ALL cell survival.
So how we targeted TBO1 in order to look at its role within cell viability was using shRNA. So shRNA, or short hairpin RNA, is an RNA molecule used to silence specific genes. It mimics natural RNA interference to block gene expression after transcription, and in our research, shRNA is designed to bind to TBO1 mRNA, triggering its degradation. So this reduces TBO1 protein levels, helping us study its individual role in T-ALL pathways.
So for our methods, first we have Western blot, where we lyse leukemia cells, ran the proteins through a gel, and then transfer them to a membrane, and use the antibody to detect TBO1, which is a protein of interest, and image the results. We also did viability staining to ensure we were analyzing the live cells. We used flow cytometry to assess live versus dead cells based on light scatter, and then we also counted and calculated the viability.
So we had to confirm that TBO1 was knocked down using shRNA before calculating viability, and that is what we did a Western blot for. So our positive control is HeLa, but the two important cell lines are JERCAT and CCRF, which are just two different leukemia T-ALL cell lines. Now the difference between CCRF, SH1, and SH2 is simply just using the shRNA to target different sequences. So using GAPDH, our loading control, to normalize the results, you can see that there is a relative decrease in TBO1 after adding the shRNA.
So then we also did cell counting to determine the cell viability. So we have our two cell lines again, CCRF and JERCAT. The white is our non-treated group, and then the red is when TBO1 is knocked down. And as you can see, when TBO1 is knocked down, the percent of leukemia cell viability or survival compared to the control is also decreased, and this represents a correlation between TBO1 and cell viability.
So we want to look at real-world applications as well. And there is a drug called tegavivint that functions similarly to the shRNA knockdown because its end result is decreasing TBO1. There's just different methods. So we tested how this drug decreased leukemia cell viability, and as you can see in both JERCAT and CCRF, after 24 hours, they're mostly significant, and the leukemia cell viability did decrease. So testing how this drug decreases leukemia cell viability makes knocking out TBO1 as part of T-ALL treatment an essential factor.
So in conclusion, TBO1 is an important target in T-ALL treatments because the decrease in TBO1 is associated with the decrease in leukemia cell viability. And this emphasizes the significance of Wnt signaling pathway in leukemia research and how treatments like tegavivint will help increase the outcome of leukemia patients in the future.
Thank you for listening.
Thank you.
Thank you. Up next, we have Rachel and Guadalupe. Rachel is from Burbank High School, and Guadalupe is from Harvard Teacher Prep. Rachel loved going to the cafeteria. This is a theme. It's a big theme this year. And she said, I loved being in the lab and meeting people who are passionate about science and looks forward to doing research in college. Guadalupe echoes some of the same things, and she was excited to see science. Well, she said what she learned was science is a balance of being strict and having fun, and looks forward to doing research in college. So get up here.
Good morning, I'm Rachel.
I'm Guadalupe, and our presentation is on regulating CD36 and FATP4 using MIR20A and MIR133A to combat Alport syndrome. So the nephron is the functional unit of the kidney, and there are an estimated 1 million nephrons per adult human kidney, and we are particularly interested in the glomerulus, which is a network of high-pressure capillaries that filters the blood.
So the filtration barrier of the glomerulus is located in the capillaries, and it's composed of three parts: the endothelial cell, the basement membrane, and the podocyte. You may be wondering, what is Alport syndrome? Alport is a genetic disorder caused by a mutation in type 4 collagen, specifically the alpha 3, 4, or 5 genes. And in the kidney, these mutations disrupt the basement membrane structure, affecting the full function of the glomerular filtration barrier. And clinical symptoms include proteinuria, hematuria, hearing loss, and visual impairment. And Alports can lead to chronic kidney disease and kidney failure, where the only available treatment is dialysis or transplant.
So we are interested in the human glomerular endothelial cells because patients with Alport syndrome exhibit elevated expression of the fatty acid transporters CD36 and FATP4, which contributes to increased lipid uptake. And increased lipid uptake is associated with mitochondrial dysfunction and damage to the cells. You can see in the first figure much more of the yellowish-greenish color, which represents CD36 and HJEC in the Alport cells. And in the second figure, you can see the red dots, which represent fatty acids only in the Alport cells. And we are interested in MIR20A and MIR133A because they are microRNAs that are known to affect the levels of CD36 and FATP4 expression through uptake. And preliminary studies from our lab indicate that MIR20A and MIR133A are both downregulated in the JEC of mice with Alport syndrome, and we think that the two may be connected.
And so we asked, do these microRNAs, MIR20A and MIR133A, regulate lipid transporter proteins CD36 and FATP4 in glomerular endothelial cells, or JEC? And we hypothesized that those microRNAs will downregulate CD36 and FATP4 expression, which would decrease lipid uptake and reduce lipotoxicity associated damage in JEC. So for the first step of our methods, we conducted cell culture. We plated glomerular endothelial cells in 24 well plates for Western blot, and we also plated them in chamber slides for immunofluorescence staining.
So transfection is a process of injecting 4 nucleic acids into the cell. In our case, we injected MIR20A and MIR133 by using a transfection reagent which damaged the cells and allowed them to pass through, and we expected them to inhibit the expression of our target protein CD36 and FATP4, and we treated them for 24 hours. And immunofluorescence is the process of using fluorescent dyes that bind to our specific protein, allowing for the proteins to be seen with the light from the microscope.
We also extracted proteins from our sample and performed a Western blot to determine if our target proteins, CD36 and FATP4, were expressed differently among our experimental groups.
So looking at the first graph, we can see a significant increase in levels of CD36 compared to the cells that weren't treated if you look at the MIR20A data, but the opposite is true for MIR133A. We actually see downregulated levels of CD36 compared to the untreated group.
We saw similar results for FATP4, with the MIR20 upregulating FATP4 and the MIR133 downregulating it. And these are the results from our immunofluorescence staining. FATP4 is shown in red and the nuclei in blue. And you can see that the results are consistent with the Western blot because the MIR20 increases the red, which is the FATP4, and the MIR133 decreases it?
So miR-20A upregulates CD36, FAP4, and human glomerular endothelial cells, while miR-133A downregulates it, which means we have proved and have disproved the hypothesis.
And we foresee that in the future we can engineer a mix of microRNAs and deliver it to the kidney as a therapy to prevent lipotoxicity, and there is possibility of preclinical in vivo studies in the future. Thank you so much to everyone at the GoFar Lab and everyone who makes LA HIP possible. And we also have a joke for you.
What do kidney researchers cash at the bank?
What?
A check.
Thank you. Up next, we have Sophia. She goes to Girls Academic Leadership Academy. Sophia says she liked the most about LA HIP was doing hands-on neuroscience research and meeting so many fun people. I look forward to bridging the gap in Latino health equity in the future. Welcome up, Sophia.
Hi, I'm Sophia Gerstein, and this is my presentation on neurodevelopmental prognosis before birth, PLX 5622 effects on primed microglia in maternal immune activated offspring. And I just want to give a shout out to Greg Dunn, who creates this beautiful neuro artwork. When a pregnant woman gets severe flu or COVID, maternal immune activation or MIA can occur. Maternal immune activation can cause fetal neuroinflammation and disrupt the blood-brain barrier, which protects the brain from the permeability to various toxic substances. MIA-induced disruptions to the blood-brain barrier can cause long-term hyperpermeability and activation of microglia around the blood-brain barrier, leading to chronic neuroinflammation. Consequently, since a recent study linked neurodevelopmental disorders with neuroinflammation, maternal immune activation is a known risk factor for those disorders. When microglia, the brain's resident immune cells, respond to maternal immune activation, they become activated and surround the blood-brain barrier. Their transformation to a reactive phenotype is marked by a reduction in branch size and an increase in cell body, alongside the expression of a cell marker for lysosomes, CD68. This brings us to our overarching research question. Does microglia depletion prevent the sustaining of MIA-mediated neuroinflammation in postnatal offspring? And specifically, can feeding young offspring with PLX 5622 diet effectively deplete microglia? To address this question, we performed the microglia depletion experiment, where my mentor Eileen injected pregnant mice with poly-IC, which was a model of maternal immune activation that induces neuroinflammation. Then those mice had baby mice, and those baby mice were given a PLX 5622 diet for microglia depletion at PD 21 after they became adolescents. And then after 3 weeks of that diet, their brains were harvested and they were cryosectioned. Then we performed immunofluorescence and fluorescence microscopy. Our results were that the PLX 5622 diet ablates microglia in the saline-injected mice without the poly-IC neuroinflammation. And as you can see, IB1 is a marker for microglia, and there's a decrease in IB1 or microglia in the PLX 5622 group. However, that change was not significant between the poly-IC neuroinflammatory experimental groups. Additionally, the percentage of activated microglia increased in the poly-IC or neuroinflammatory group. And as you can see, CD68 marks for microglia activity, and there's an increase in CD68 in the poly-IC experimental group. Ultimately, the PLX 5622 treatment depletes microglia in the saline groups, and the poly-IC injection significantly increases the percentage of activated microglia. However, since the PLX5622 treatment inhibits the CSF1R pathway, the lack of effectiveness of the PLX5622 treatment on the poly-IC neuroinflamed experimental group may be associated with downregulation of the CSF1 pathway. Future research should investigate the role of the CSF1 pathway in microglia activity and include more experimental trials on mice. Thank you so much for the Bonin Lab for all your support.
Wonderful. And last but absolutely not least, we have Jonna and Valeria. Jonna goes to Amino Inglewood Charter High, and Valeria goes to Harper Teacher Prep. Jonna says that she liked most about LA-HIP was the supportive lab environment and her supportive mentor. Valeria says that she loved most about LA-HIP was the cell culture and is looking forward to joining a research lab in college. Welcome up, ladies.
Hello everyone, my name is Valeria.
Hi, my name is Jenna.
And we're studying the effect of novel ARCH20 trip mutation on PAX4's transcriptional function. MODY, or maturity onset diabetes of the young, is a rare type of diabetes present in less than 5% of the diabetic population. Unlike common types 1 and 2, MODY appears by the age of 25 and is a result of a single mutation that impairs insulin secretion. A variety of mutations are associated with MODY. Therefore, patients have unique symptoms that are complicated to diagnose and treat. MODY9, or MODY type 9, is associated with mutations in the PAX4 gene. Mutations in the PAX4 typically lead to developmental defects in beta cells and can result in immature beta cells and low insulin production. PAX4 is a transcriptional repressor that plays a crucial role in beta cell development. In mature cells, PAX4 switches its role to regulating blood sugar levels, such as repressing uncontrolled insulin production. Therefore, mutations in this gene can result in poor beta cell differentiation and insulin secretion.
A recent patient at CHLA was presented with the novel PAX4 mutation in the 58th nucleotide, leading to a single amino acid change in the binding domain of PAX4, which is responsible for recognizing and binding to its target genes. In silico analysis, we predict that this mutation is pathogenic and will disrupt PAX4 from binding to its targets. However, the clinical significance of this mutation and how it contributes to the patient's diabetes is currently unknown. Therefore, we aim to determine if this ARCH20 trip mutation would impact the molecular function of PAX4 as a repressive transcription factor. Since PAX4 is a known repressor of insulin in adult cells, we will use the insulin promoter as a quick tool to determine if PAX4 is binding to its transcriptional targets. This leads to our hypothesis that the novel ARCH20 trip mutation will impair PAX4's function as a transcriptional repressor when being expressed in Min66 cells.
And so Min6 cells are a mouse pancreatic beta cell line that secrete insulin and do not express endogenous PAX4, making them a suitable choice to study insulin activity. We utilized a CRISPR-engineered Min6 cell line called Min66 that is integrated with an insulin luciferase reporter in which luciferase and insulin are secreted in a 1-to-1 ratio. We seeded our Min6 cells and then transfected them the following day using lipofectamine to induce PAX4 wild type or PAX4 mutant expression. We used Min6, integrated Min66, and wild type PAX4 plasmid as a control for this experiment. To determine if PAX4 mutant is binding and repressing the insulin promoter, we collected media for a luciferase assay 2 days post-transfection. We then measured luciferase expression on a luminescent plate reader. To determine if our Min66 cells express PAX4 protein variants, we fixed our cells for immunocytochemistry, tagged them with PAX4 antibodies, and imaged using a fluorescent microscope. So here we have our results for the immunocytochemistry, and here we confirm that PAX4 is expressed only in our transfected Min66 cells, which are featured on the 2 bottom rows. The top rows are our controls, and as you can see, they have no PAX4 expression expressed by the red.
When we analyzed luciferase expression to determine if PAX4 is binding to its target, insulin, we saw a difference in the luciferase activity in the mutant PAX4 when being compared to its wild type. The PAX4 wild type protein repressed the insulin promoter as expected. However, the mutant protein failed to bind to the insulin promoter and therefore did not repress insulin. There was no difference in insulin expression in the PAX4 mutant sample when being compared to the non-transfected control for Min66 cells. Based on our experiment, we confirmed that PAX4 expression through immunocytochemistry and analyzed its transcriptional function using an insulin luciferase reporter as a tool. The PAX4 mutant protein failed to repress insulin expression in the luciferase assay, suggesting that PAX4 is not binding to its targets and supports our hypothesis that the ARCH20 trip mutation is impairing PAX4's transcriptional function. We believe that this mutation is primarily contributing to the patient's diabetes by failing to bind to the transcriptional targets required to differentiate mature beta cells. In the future, the Georgia lab aims to study the PAX4 variant during pancreatic beta cell development to determine if it binds to the transcriptional targets required to differentiate mature beta cells. And we would like to thank the Georgia lab and the programs that funded us.
Thank you for your time.
Now, would you believe me if I told you that before you guys got here, they were all singing like karaoke on this microphone? Because the shift is incredible. Specifically work by Rihanna, if you're familiar. But like I said, they have been nothing but a joy to work with. We're going to have a couple closing remarks, one from Dr. B and then Dr. Fry before we go out to our reception. So I'd love to welcome up the man, the myth, the legend, Dr. B.
Good afternoon, and thank you all for coming. I remember when I was in charge of this, I was more nervous than the kids. Anyways, I also know you guys are all hungry, okay? So I'll make it very quick, okay? But when I thought what I want to tell you guys, 20 years ago, I proposed the idea. You guys weren't even born. And Dr. Fry probably was still in college, okay? Anyways, you heard already all the thank you for the program. I am particularly proud of that after 20 years, the program still goes very strong. This is because of the leadership in the program and the tech pad and the support from the hospital. Give me a hand, please. Unfortunately, the current environment has changed. And science and scientists are really under attack. And I have to say, I can only hope that this is going to change. And who do you think I hope for? You guys. You're going to represent the next generation of scientists and physician scientists and MD PhDs. You got to stay the course. You got to maintain your interest in science. We need you guys. It can't go on like this, and it will not go on like this, what we experience. But we need your support. I conducted a book club with the students, and we read a book by David Quammen on emerging infectious diseases. Now, Flo told me it was a very boring book club. Just kidding. Just kidding. But I asked the student to write some sort of a summary. What was it? What was interesting for you? And almost everybody said the important part that they learned is how critical and crucial it is to have science communication to the public. And you probably realized how difficult this is, because some of your slides, if you're a layperson, are difficult to understand. And it is crucially important that we as scientists, and you as the future scientist, gonna learn how to do this, so that the people who are not scientists actually understand what you're talking about. It is not easy. And we as scientists who are older, we have the obligation to teach you how to do this, okay? I also told you in the book club, and I feel strong about this, you were the lucky guys who get into the program. There were 200 applicants, and about 10% made it. You were the lucky ones. And with that comes an obligation. You have to go and be the messenger to your friends, to your school, how fun science is. I want you to go and tell them that you had fun in the lab. It was not nerdy. No, it's not. And the whole labs are full of people from the entire world, all over the globe. And it is something that you're going to have to communicate. You can't just go and keep it for yourself. I will hold you guys accountable. Okay? I will hunt you down. I have your email. Okay? Does anybody know who Mr. Wang is? Jensen Wang? Anybody know? Anybody know what NVIDIA is? NVIDIA, Jason Wang is the CEO of NVIDIA. NVIDIA is this incredible company that makes these sophisticated computer chips that you need in AI. I saw a video clip on Instagram, and yes, I do have an Instagram account. Okay. He was talking to graduates from Stanford a couple of years ago. And he says that students who go to these elite schools often have very high expectations. Fancy job, good salary, this, that, my career is done. But he says his experience with students from these institutions have very low resilience. Resilience means I stay the course. I dig through this whole thing. It will be challenging. You go to the fancy universities and colleges in a year from now, and it's gonna be hard. But you gotta go and grit through the hard time. That is required for success, okay? And I have the confidence, since you arrived, LHIP, you know, I have the, despite Dr. Fry, I have the confidence that you're gonna stay the course. And again, I will hold you accountable and haunt you down, okay? All right, enough is enough. My last thing is I asked the students, so what was the best aspect of LHIP at the summer? You know what the answer was? Flo said the food in the cafeteria. Anyway, thank you all for coming. Right.
Now, I will say that in the orientation session, when we talked about how they were going to have meal cards for the cafeteria so they could all have lunch together, and they're all looking at me like that's a benefit, and I'm like, no, really, it's good. It's a good cafeteria. Now you know, it's a good cafeteria. Thank you, Emil, both for founding this fantastic program and for those important remarks and words of wisdom. This is a tough time to be a working scientist. It's also a tough time for programs like ours that are committed to having the scientific workforce look like the population of California in the future. So one request I would make of all the students is that if you had fun in the program this summer, go back and tell your friends who are going to be juniors next year about it. You can probably imagine that there are some constraints on how we can advertise the program at the moment, and we don't want that to change the mission. And we're not going to let it change the mission, but we need your help in spreading the word that we're still here and we will still be here. We've outlasted a lot of things, and we're going to outlast this. So if you would do that for us, that would be fantastic. I don't want to end it on a down note, so I'm just going to reiterate that you guys did amazing work this summer, really terrific work. You picked up all the science really fast. You learned so many things in the lab. I hear all the time from mentors how much fun they have working with you. I also hear all the time from professional working scientists who come to the symposium about how good your presentations are. I've had several people tell me they wish their graduate students presented this well. I'm not making that up. That's several people have mentioned that. So you guys really have done great work. If you have even the tiniest spark of enthusiasm for this business as a career, fan that flame. It really is a lot of fun doing this. It is, sorry Emil, but it is nerdy as heck, but it's the fun kind of nerdy. And it really, I frequently have students ask me, well, if I couldn't do this, what would I do? And I don't know, because I think I have the best job in the world, and it's not easy, but nothing worth doing is. So if you're interested, keep at it. As Emil says, stay the course, have the resilience to power through, and you'll find that it's extremely rewarding. All right, that said, let's go eat.